Day scientific seminar. If anyone's still out there grabbing coffee, we'll give them a minute to walk in. It is my pleasure today to introduce Dr. Pete Guiden, who's here with us from Hamilton College and is going to talk about the agents of mouse destruction, which is a very exciting heading. Pete was a grad student with Winslow who had to leave for another meeting, unfortunately, and has been at Hamilton College since 2021, where he's an assistant professor and has really been very prolific with leading students in some really exciting research, looking into plant-animal interactions and influences of changing climate and global conditions on plant-animal interactions and their consequences of that for ecosystem function. So thank you all for coming, and help me welcome Dr. Guiden.
Thank you. Yeah, thanks everyone for being here. What a pleasure it is. I've always been, aware of all the excellent work that's been going on here at Cary, since I was a grad student. And the fact now to be, just a couple hours down the road from all of you and to come here and meet you is such a pleasure. So thank you so much for having me. So I'll start real quick by just sort of acknowledging, research isn't done in a vacuum, right? I strongly believe it's a team activity. So a lot of the work that you'll be seeing today is sort of describing work that stemmed or grew out of my graduate program, where I was working with Dr. John Orrock and our postdoc in the lab at the time, Dr. Brian Connolly. And then we'll also acknowledge some of the postdoc work that I did with Dr. Holly Jones at Northern Illinois. And then increasingly, a lot of the talk will involve sort of the lab that's going on at Hamilton right now, me and my students, and so sort of the most recent projects that we're getting off the ground now. So I thought it appropriate to maybe start with sort of, addressing the sort of fundamental questions that I'm going to be talking about today. So the big one, the sort of overarching thing that I think about in terms of my research is this idea of can we understand communities and why communities are structured and why they function the way that they do, and the species interactions that they contain by sort of thinking about, okay, what are the different consumers doing? How are they behaving, right? So that's sort of one way that I try to address my research is integrating these ideas of community assembly and behavioral ecology. So I'll talk a bit about that. And then the bulk of the conversation today I'll sort of center around one of my current research projects, which is looking at these seasonal changes in habitat structure. So what happens in northern temperate forests as the seasons progress, and how does that change consumer behavior? And specifically, what are some things that people are doing in terms of things like climate change and proliferating invasive species that are maybe shifting these seasonal interactions? And then finally, I'm an optimist. Chicago Cubs fan, so I've had to be optimistic throughout my whole life. So I'll talk to you about, okay, what are some ways that we can maybe hopefully apply this understanding of habitat structure and rodent behavior to improve restoration ecology outcomes, right? How can we rebuild the wheel perhaps a little bit better? So we'll start off with sort of a background dive into this idea of community assembly, optimal foraging behavior, behavioral ecology. How do we sort of make these two ideas talk to each other? So that's just sort of a brief overview, right? So specifically with the community assembly theory, I'll talk to you a little bit today about specifically this role of biotic interactions, right? And so what do you do if you have two closely related consumer species that actually might prefer to consume different food sources in their habitat? And then second of all, we'll be talking about optimal foraging theory and how do the decisions that these rodent foragers make, specifically about when they're active and where in their habitat they're using space, how does that influence the outcome of these trophic interactions, right? And ideally, can we sort of synthesize or integrate these two ideas? So I'll start just to sort of get us all on the same page. Hopefully this isn't, totally news to a lot of us, but this is my sort of, Fisher-Price, my first community assembly framework that I show my intro classes, right? So if you start with a regional species pool, the first big filter that sort of dictates whether or not something's going to show up in a community is, can you actually reach that site, right? How important are dispersal limitations, right? And so some of these species will get lost because they simply can't pass through that dispersal filter. They lack the ability to disperse to a site. And then second, species are going to bump up against this environmental or abiotic filter, right? So maybe there is some extreme heat or cold conditions, maybe there's a dry spell, and the species can't really overcome that. And so in this case, those green species would be filtered out here. And so finally, that leaves you with a smaller subset of species, and then there might be some biotic interactions. So things like seed predation that I'll talk a lot about today, or herbivory, or disease. And so finally, we're going to lose the square species according to that filter, and you're sort of left with this subset after everything has been filtered out, right? But learning sort of where along this process exactly are species getting filtered out is actually a really challenging task, right? And so a lot of my work has to do with this final step, these biotic interactions, and trying to understand if you are in a community with a whole bunch of, for example, voles, one type of rodent, or a whole bunch of Paramiscus species, and this other type of rodent, an acorn, which one of those are you more likely to survive in, right? So that's one example of how this looks in the real world. And I'm fascinated by seeds. So if you look at sort of like a really generic plant life cycle here, right? So you've got seeds, and then they germinate into seedlings, and then seedlings eventually grow up into a mature plant. I'm fascinated because a lot of the filtering happens really early in the plant's life cycle. So something like, if you look at this global meta-analysis done by Angela Moles a long time, like 20 plus years ago now, something like 5% of seeds germinate, right? And so of those seedlings that arise, only about 6% of those are going to recruit into mature reproductive adults. So what that means is that on average, and this is a huge sort of like back of the envelope calculation, something like 3 out of every 10,000 seeds is going to actually survive to a reproductive age. So the vast majority of seeds and seedlings are getting filtered out. So understanding which of these filters are stronger at a given time or place, that's really vital information for community ecologists. But this is easier said than done, right? So it's one thing to like understand that in an abstract sense, but the really frustrating and cool and exciting aspect of this is that context dependency really drives what these filters look like, right? So for example, one of my recent favorite examples of this context dependency is if you consider sort of a generic interaction, tritrophic interaction here between carnivorous birds, caterpillars, and trees. In a regular year, this interaction looks something like this in this food web, where the trees are getting this indirect benefit from the carnivores because they're eating the herbivores. But in a big cicada emergence a year, like the one that we were recently having, so suddenly 80-some species of carnivorous birds are going to stop primarily eating caterpillars, and most of their diet is now going to shift over to emerging cicadas. So suddenly the rules have changed. The filter strengths have changed, right? And so that really complicates this, right? Another example, since I'm here at Cary, I'll talk about Jacqueline Schnurr's paper. I love to cite this one every time, right? So this is looking at seed removal in the forest floor. And so higher values here essentially mean more seed survival. And you'll notice that like the neighborhood, the spatial location that you end up at as a seed, if you happen to fall in a sugar maple stand or if you happen to fall in a red oak stand, dictates the strength of the seed removal filter, right? So if you're in a red oak stand, you tend to have much more activity and seed removal from those seed predators. And so your likelihood of survival has just dipped, right? And so for a long time, folks have written now about how understanding this context dependency is a real frontier, it's a real challenge for community ecologists, right? And so understanding these mechanisms and how the whole complicated system interacts and what the different pieces are, that's a big part of what I'm thinking about when I wear my community ecology hat. But I mentioned that that's just half the coin for me, right? So on the other hand, I also came from a lab where we would think a lot about behavioral ecology, and specifically how do rodents choose to behave, right? So what are the choices that these rodents are making? And so the big umbrella theory for this that we talk about is the optimal foraging theory. And it sort of has this golden rule, right, that rodents are really any sort of consumer. They should forage, they should be active in times and places where the benefits of that foraging outweighs the cost, right? So you need to make more than you're spending, right? Which makes sense. And so the benefits in this case would look like things like, calories. Are you getting enough calories? Maybe an important micronutrient like sodium or something, right? Whereas the costs are things like predation risk. Is it free to forage, or are you foraging in a really risky habitat where an owl is going to swoop down out of the sky and eat? That's a really high price that you are not going to want to pay, right? But then there are also things like metabolic costs, right? And missed opportunity costs. Maybe if you're out foraging, you're not defending a nest or a territory, right? And so primarily, I'll be talking about this predation risk cost, but a little bit about these metabolic costs too, right? And so the way that we might think about this optimal foraging theory would be running something that we call a giving up density study, where you put a depletable food patch, out in your habitat, and oftentimes they're paired in a safe habitat and a risky habitat. Everything else is held constant, and you look at the difference between those two and say, okay, that can dial in this case, the cost of predation risk is whatever the density difference is there. So a higher value on this y-axis would indicate that there's less foraging activity, more seeds have been given up on, right? And so usually these safe patches are more depleted, right? In this case, Ken Schmidt ran a study where he crossed this safe risky treatment with different secondary compound defenses from different plant species. So for example, if you coat the seeds in a toxin, then suddenly the squirrels stop eating that much faster, right? So the way that I think about optimal foraging theory is to try and understand what are these safe and risky times, what are these safe and risky places, right? So there's a spatial dimension to this fear component, but there's also a temporal dimension. So the spatial dimension involves, okay, so where do predator and prey overlap, right? Where do they co-occur? And those are some of the most risky habitats. But also, a lot of times we see micro-habitat structures that make a predator either more or less lethal, right? So for example here, imagine you've got 2 mice in 2 different habitats. And the habitat on the left has a really sparse understory, right? It's relatively open. There's a few shrubs, but not many around. Whereas this other really densely grown habitat has a lot more nooks and crannies for that predator to hide. Perhaps, if again you're imagining an owl swooping out of the sky, you think, oh yeah, the owl's probably not going to try to hunt in here because that's a lot harder, right? Whereas this open habitat is going to be a lot better hunting. Right? So in this case, the owl is going to be more lethal in the habitat structure where it's more open, right? Whoops. Oh, there we go. Okay, but I also mentioned that there's this temporal component as well. So the temporal component often involves these activity schedules or activity budgets. And so predators and prey, they're not only need to encounter each other, they not only need to be present at the same place, but at the same time, right? And so there's a lot of windows in time where predators and prey might be more or less likely to overlap. And one of these, if you study rodents, has to do with moonlight and lunar cycles. And I have to say, when I was a graduate student, I started reading these papers and I was like, there's no way that's true. Like there's no way that the rodents are actually paying attention to the moon. And I talked to my friends that were studying, fish dynamics and predatory fish. They're like, yeah, no, it's all about, lunar cycles and moonlight. And I was like, okay, so maybe there's something to this, right? So it turns out that a lot of the big predators for rodents, especially when you're talking about raptors, for example, visual hunting is really important. These visual cues while they're hunting matter. And so times or places where a lot of moonlight is coming through means easier hunting for them, right? And so if a rodent, you can imagine, is maybe going to be active all night long, well, suddenly at the point in the night where the moon is at its brightest peak, then that's where that overlap between predator and prey is not only going to be greatest, but suddenly that predator is more lethal, right? So we would expect that if you're an optimally foraging rodent, maybe this time of night is one where you want to lay off, right? So that's maybe not the best time to be active. So bringing this sort of back together before I move on to the real nitty-gritty of the research, I want to sort of lay the groundwork for one of the studies I'll be talking about with invasive species later on. So I want to point out now that both of these theoretical frameworks can and have been used to sort of understand different threats to biodiversity and how communities and ecosystems understand that, right? So if we're thinking specifically about invasive species, when we think about community assembly and why invasive species spread and why they get here, people have posited for a while now that, well, maybe one of the explanations here is that these invasive species are more likely to pass through all of these environmental filters, right? So they're more likely to disperse because people are giving them a free ride around the world, right? Where they have global distributions now because they're hitchhiking or being intentionally introduced somewhere, and that's that dispersal filter. But additionally, they might have traits that make them more able to cope with environmental stresses, or they might be less preferred by seed predators, or they might lose diseases. And so they're more readily passing through those biotic filters, right? That's sort of one of the prevailing wisdoms when it comes to invasive species management. And then also, optimal foraging theory plays a role in plant invasions, right? And so a lot of times what we see is that as invasive plants establish and spread, they often co-opt native animals, native wildlife, to sort of help further facilitate that spread. And so one way that they do this is they invade, they start to grow in the habitat, and they radically restructure that habitat, either by providing new food resources, changing the habitat structure and what that looks like. And then in turn, that induces some sort of behavioral change in the native wildlife. Maybe it changes how birds are nesting, or what rodents or deer are eating, or how insects complete their life cycles, any of these things. And then eventually that should scale up and have a community and ecosystem-wide impact. So I just want to point out at this point that both of these theories have been sort of invoked to explain this phenomenon of invasive species. But thus far, there hasn't been a lot of work to maybe sort of integrate these two. And that's sort of what I'm hoping to address with some of my research here. So my last bit of background, I'll just talk about, why rodents, right? So I actually started with a botany master's degree. I was studying invasive honeysuckle and how it was spreading through white-tailed deer. And then I joined John Orrock's lab, and he was able to convince me that, hey, rodents are actually pretty cool, right? So they are, I like to point out, they're the most diverse order of mammals on Earth, right? So there are lots of different interesting species doing interesting things in the community and the ecosystem. On that note, they're actually pretty central in most food webs that we think of, right? They're doing a lot of the consumption. They're eating things that we care about, like invertebrates, like seeds. And in turn, they're also being eaten by a lot of other things that we care about. Things like, endangered carnivores are eating rodents. What also makes them interesting in this food web context is they tend to have pretty flexible diets, right? So more on that later. There's also a really strong connection to human welfare and human health, right? So lots of emergent diseases have their origins in rodent communities. And on that note as well, a lot of these rodent communities are actually being homogenized and simplified, right? So a lot of rodent species, especially here in the Northeast, we're losing those sort of like interesting species like flying squirrels and jumping mice, and you're essentially left with a community that's enriched with deer mice or white-footed mice and chipmunks, right? So there's been a simplification and a homogenization of those communities. So what does it mean if we lose that biodiversity, right? That's the question that I think is still pretty open, right? And there's sort of this classic view, I think, that because rodents have flexible diets, if you look at their sort of jaw structures, you say, okay, they're flexible, they're adapted to eat a wide range of food sources. For a long time, ecologists have essentially treated rodents like, in many ways, they're substitutable, right? But especially in the trophic interaction literature, there's recent evidence suggesting that this might not be the case. There's a little bit more nuance than that here. And so we're seeing, in fact, a lot of interspecific variation in even really closely related species in what they eat. So for example, here's a study from about 10 years ago that Michael Kramer was running up in northern Wisconsin looking at Paramyscus leucopus and Paramyscus maniculatus, who, if you had the two of them in your hand, you'd have to look really closely to tell which of the two species you're actually looking at. And in fact, what he found was that the Paramyscus leucopus, pretty much for the most part, they show a preference to mostly eat sugar maple. Whereas Paramyscus maniculatus, this really closely related, morphologically almost identical species, is actually eating only red maple. And this is a problem because in a lot of times when people are writing up studies, it's common to maybe even lump those two species together, when in fact we're showing there's actually a pretty subtle but important and consistent difference between the two of them, right? So again, this gets back to that idea of like, what is that biodiversity? Is there actually some cryptic biodiversity there that we really need to appreciate? And even if you look within one single species, right? So in the case of like only looking within Paramyscus maniculatus in this case, Alison Brem, who was working with Alessio Mortoliti at the University of Maine for her PhD, ran a series of studies looking at rodent personalities, so repeatable, consistent behaviors that the same individual will exhibit time and time again. And so she assigned them different scores on traits like timidness, like how likely they were to go into an open field and explore, or would the mouse like hang back at the back of the trap and be wary of going out. And it turns out that in this case, this trait, the less timid you were, the more likely were you to take a seed and cache it and actually give it a dispersal, basically a mutualism, right? So you're actually dispersing that acorn, burying it so that it has a chance of regrowing. Whereas if you were a really shy individual mouse, then you were actually more likely to just sit there on the spot and consume that acorn, right? So species to species, we see subtle differences. Within one species, we're starting to see subtle differences, right? And so this kind of begs the question, like perhaps maybe by better understanding these differences in behavior, this might provide an important piece of that context dependency in how the community is structured, and we can maybe better figure out what these levers are. So all that to sort of set up, this idea of, okay, now I'm going to tell you a little bit about what we're doing to examine this in the context of seasons, right? So how does consumer behavior change seasonally? Does it matter if we study that? And how are people changing seasonal dynamics, right? So let's raise your hand. Are you guys native to the Northeast? Raise your hand if you grew up here. Grew up somewhere else, raise your hand. Okay, cool. Can we all agree that the Northeast is awesome in the fall, right? So like the summers are glorious, right? It's like 70 degrees here in New York. I love it, right? I melt in the heat, So like this time of year, I'm psyched, right? Like the fall colors are changing. It's beautiful. People come from all over to see it. And then, winter has its beautiful times too, right? like hopefully nice fresh snow. We all enjoy that, right? And so what I just want to appreciate in showing these pictures and sort of like evoking this in your mind is that these habitats look really different, right? Like one habitat, these are all just shots from the Adirondacks, but the habitat structure changes tremendously. And so animals have to cope with that, plants have to cope with that, right? And so a lot of what we do as ecologists happens to be studying, what's going on in the spring and what's going on in the summer. And you guys are very lucky, right, in the sense that you're maybe not held to an academic calendar. When I'm teaching 3 courses in the fall, it's hard to get out there in the fall and measure what's going on, right? Spring semester or winter semester, there's a lot of other things going on, and so we tend to mostly just look at what's going on in our systems in these seasons when the world is green. But for point of argument, what if there's something really important happening in the winter and we're just not ever looking at that, right? Like what if there's something important happening in the fall, but we're just often too busy to get out there and measure it? That was sort of like a central question that I was asking when I was a graduate student at the University of Wisconsin and sort of guided my research today. So I'm going to talk a little bit about some of the work that I've done on winter habitats and how they're changing. And then I'm going to talk a little bit about fall habitats and how they're changing, right? So we'll start with winter. Just bear with me here for the sake of how the story will unfold. So winter, we tend to think of it here in the Northeast, snow, right? Snow, lots of snow hopefully, but increasingly that's not the case. And in fact, if you look at sort of the climate change projections broken down by season, right, winter is one of the fastest changing seasons in this neck of the woods. And so here's just a figure looking at snow cover anomalies versus I think a 1980s baseline here. And as you can see, as time has gone on, more and more of these negative values here. So snow cover is becoming less and less extensive and common over the last few decades. So these boxes are broken out. Here's the blue year sort of broken out into a map of March snow cover. The percent of days in March that had snow cover across the northern hemisphere. Same thing with this red one over here. And I really like this illustration because what it highlights is that if you are, Winslow's not here, so I'll pick on him. If Winslow's in Alaska, in March, it looks the same, whether you were in the 1960s or today, right? But if you're hanging out, closer to home here, these are the areas, right? So sort of like the low latitude, mixed deciduous forests of the US, also, Mongolia, northern China, and Western Europe. Those are the places where snow cover is becoming basically absent in March, right? So this area of the world is showing some of the fastest responses to climate change happening actively. And that is a fundamental loss of a snow-covered system transitioning to a snow-free system. So another way to think about this, here's my completely made-up graph of data just to sort of illustrate the point, is historically, what did a northern temperate forest look like during winter? Well, maybe you have these sort of early snow events, early in winter, and then they melt, and then you get a little bit more snow, but then it's really cold, so instead of disappearing, that snow is just going to keep accumulating, right? Until eventually we get to spring, everyone's really happy in April, the snow starts melting, it's 50 degrees again, and we're wearing shorts. Great. But the sort of new normal that we're seeing is that it's hovering right around that freezing 0 degree Celsius threshold. And so instead of accumulating, we're just getting a lot of this, like, not only less snow, but the coverage of snow is really decreasing, right? Because you're just getting a lot more of this back and forth winter whiplash. And so we go long periods of the winter where we fundamentally lost this snowy cover, right? So what does this do? I'll show you some research now that I was conducting as a graduate student. So from the community assembly perspective, one of the things that this does to especially plants who are stuck, right? They're stuck still. They are used to sort of having this protective snow cover around them. Well, there's maybe now a new abiotic filter, right? Where now in a snow-free world, you're exposed directly to the cold and variable winter air, right? So we're able to show that in a bunch of different woody seedlings, if you expose them to like an extreme, for in this case it was minus 16 degrees Celsius, cold event that would follow on the heels of a warm spell, then you get a lot less growth in the following summer. Right, so those seedlings were a lot less likely to grow fast, right? And so essentially this is, again, you're looking at the world during the growing season, and you need to appreciate that, hey, actually this is explained by something that happened in a previous season where we typically don't look. And on the other hand, abiotic filters we can maybe kind of wrap our heads around, but the biotic interactions are extremely variable, right? So like how does the loss of snow actually impact things like trophic structure of communities and ecosystems? There are so many complicating factors, it's really hard to figure this out. So one study that we looked at, I don't know if this is really smart or really stupid as a grad student, but I did a snow removal study out in the woods where I would go out and shovel snow out of the forest, right? I'm not gonna do that in my mid-30s now because my back can't handle it after having 2 kids. But so I went out in the snow and we shoveled a whole bunch of snow to sort of simulate that loss of that snowy cover. And one of the things that we found, and we measured seed removal, sorry, I should say that, after removing that snow. And so one of the things that we found was that where you remove that snow, you would occasionally see actually increased survival of the seeds. So they were less likely to get consumed. But that only occurred at sites with lots of woody debris. So this suggests that there's something about the way that the consumers are using their habitat and the context of this microhabitat structure and the availability of woody debris to hide behind or travel on that influences how climate change might change this trophic interaction, right? So it's really complicated, right? There's a lot of complicating steps here. And so that suggests that we maybe need to bring in these ideas from optimal foraging theory to better understand that, right? So one thing we might think about is I mentioned those metabolic costs, right? So as we lose snow, we see organisms are increasingly exposed directly to air temperature. So here's a figure looking at winter air temperature, the ambient air shown in dark blue here in northern Minnesota. And then under a shallow snowpack in the light purple, and the light pink under a heavy snowpack of about 40 centimeters. And so what you notice is not only is that cold air really cold, but it's also variable, right? It's whipping back and forth, right? It's a lot of uncertainty for the organism about what is my environment going to look like tomorrow. But under that nice, cozy snowpack, it's not only warmer, it's more stable, right? It's more predictable. So perhaps as we lose snow, it's going to impose greater metabolic costs on organisms. Additionally, this snowpack represents an important predation refuge, right? A way that you can actually hide from your predators during the winter, right? Rodents are really good at tunneling through the snow. Like some of you might have seen, voles staying active through your yard all winter long, sort of grazing these highways, these little tunnels, right, in your grass lawn. And so losing that snowpack, you might also be more exposed to your predators. So we studied this. We actually had an interesting idea after reading some papers that pioneered this to put out some Subnivean camera traps for one of my PhD chapters. So we wanted to put some camera traps out in the woods in essentially a bucket with a motion-activated camera facing down on the forest floor to see, okay, who's using this winter habitat where we're out looking, right? And so here's one of the first things that we noticed was that Peromyscus leucopus, the most common species in the forest, and then Myodes gapperi, the southern redback vole, common but not dominant in the community, we've plotted their activity as a function of time, and we also looked at sort of what the snow and temperature conditions were doing over that same time. So here you can see this is actually a pretty typical winter where we have that gradual accumulation of snow, right? So sort of like a historical conditions sort of thing. And we noticed that right around 30 centimeters, I'm not showing the data here, but I'm happy to show you later perhaps if you would like. Once you hit about that 30-degree mark, the temperatures got really stable and hovered around -3 Celsius. Whereas down here, we're seeing soil temperatures anywhere from like -6 to -10 and much more variable. So what we actually see is that the Peromyscus leucopus, these mice here, they are essentially more active in this early, more unpredictable, less snow-covered time of the year. But then we start to only see the meiodes pop up once that nice, deep snowpack has established, right? And we thought, oh, that's really weird. Like, are the Peromyscus, like, are they dumb? Like, do they not know that it's cold and snowy and they're not waiting? And so we're like, well, I wonder what's happening there. And then I had the idea to actually look at the timestamps on these photos and see like what actually was going on. And so what we found was that Peromyscus leucopus, which is pretty much, like I think most people would agree, pretty much 99% a nocturnal species, under even that shallow bit of snowpack, they're actually active during the day. So they're awake during the day underneath the snow. They're out and about. They're taking advantage of whatever daytime warmth there is, and they're avoiding these nighttime areas where it's cold, right? So they actually do, they're not dumb. They know what's going on, but they're just totally reorienting their behavior to take advantage of the fact that, okay, I've got snow overhead, so I'm not super visible to my predators, and I'm going to be active during the daytime to sort of minimize that metabolic cost. Whereas meiodes has a different strategy entirely, which is just wait until you have that really deep snowpack, and then I can be active sort of whenever, sort of have a uniform activity pattern. So that was really surprising and really interesting to me. So I thought, okay, rodents are doing weird things that we don't really think about a lot. And I wondered, okay, what are sort of the trophic interactions looking like? That's sort of my next question, right? Is like bring it back to sort of understanding these community level phenomena. And so this is one of the first studies that I did with a student at Hamilton that I'll talk to you about now. And so we wanted to look at, okay, are there sort of these seasonal patterns in the number of seeds that rodents eat? Are they changing how much they eat sort of throughout the course of the winter? Are maybe different seed predators showing up at different times? And are they maybe switching seed preferences throughout winter depending on how cold the air temperature is or something like that? And so to study this, we deployed our little foraging stations that I'll tell you about in just a minute at these 14, we recently launched them long-term. Can I call them long-term yet? It's been 4 years. They're going to be long-term someday, right? But it's a budding long-term monitoring site with me and a couple of the other faculty at Hamilton in our experimental forest. And we put seeds out to see what the rodents would prefer to eat. We had 2 native seeds and 1 non-native seed. So the 2 native seeds were sugar maple seeds and eastern hemlock seeds, and then invasive honeysuckle for the non-native. So all common species that we find in the different habitats around our forest, right? And so Maddie Vavra was my student collaborator on this project. They're excellent. They're now a PhD student in Rebecca Rowe's lab at New Hampshire. And so together they and I came up with this idea of this temporal rodent activity, seed predation, and herbivory camera, also known as a trash cam, because it is just a trash can that we put out in the woods, again with a motion-activated camera. You drill a couple of holes in it so the rodents can get through, and voila. Maddie actually made a lot of progress on my first really, sort of half-baked attempt on this during my PhD, where they figured out that if you in fact take like a reading glass lens, like a cheater lens, and kind of grind it down with a Dremel and tape it over the lens, you get a super focused, like crystal clear picture of what's going on there. And so that greatly improved our ability to sort of differentiate the different species. And so you can see that this is a really clear photo, just sort of pointing down at the tray of seeds. And so normally I should sort of pause here and impress the fact that like for most of my experience putting seed predation studies out, it's a lot of, you take like a 1-gallon ice cream bucket, you put it out in the woods with some sand and like some options of seeds, and you come back and you say, okay, like 10 seeds disappeared. I'm going to assume that that's sort of like proportional to whatever we're trapping and whatever we've quantified the community to be. We're going to sort of assume that everyone's sort of equally likely to go in the seed bucket relative to sort of what their abundance is, right? But the advantage of this is like we can actually catch them in the act, right? It's sort of like a Ring doorbell for a seed predation setup, where in this case, you can see that that's a Paramyscus species. We actually can't, this is the one drawback, we can't differentiate based on the video alone between the Maniculatus and the Leucopus, so the two species that we do have in our forest. And we can see, sorry, back that up one more time. And so again, this little guy came in and he went right to, he or she, we actually don't know that either, but they're immediately going over to those honeysuckle seeds. And they're gonna scoop that up and eat that first, right? And we actually see, okay, how long were they in there? Did they just go in there for 10 seconds? Did they camp out in there for an hour? And we can actually understand on a species by species basis which of these species are going in to consume different species of seeds. Like that's pretty cool, right? So again, we're getting more to linking the behavior of different species to this community level phenomenon of how many seeds get eaten in the woods. So I'll show you some data here. This is in revision now at Ecosphere. And so what we're going to look at first is basically just the number of visits. How many times did our Ring doorbell catch different species in different months, October, December, January, April. We put them out for about 1 week in each of these months. And we actually found only 3 species going in. So mice of some sort, red squirrels, and chipmunks. And so what we see is that actually red squirrels really liked our foraging stations, both in October and in April. But there's sort of a switch that happens during winter, where we actually see that in December, it's mostly mice, and in January, it's about an even mix of the two species, right? So just sort of over time, we get the pattern that, maybe red squirrels are doing something else in the heart of winter, but that's when the mice are coming most likely into our bucket. So there's maybe a little bit of a seasonal switch there, but nothing super dramatic, perhaps. And here's the figure that I think is the most cool out of this paper, the one that I'm most excited about. If we look at the proportion of seeds removed in a tray, so like, we had 18 seeds of every species out, what proportion of those got eaten by deer mice, chipmunks, and red squirrels? Because that's really what we wanted to do, right? We wanted to break it down by species. And we've got our 3 species of seeds here, sugar maples in yellow, honeysuckle in green, and the eastern hemlock in blue. So what we actually see is that both deer mice and the red squirrels kind of equivalently are like equally likely to pick up a sugar maple seed. Chipmunks, we saw 1 chipmunk go in, so we're not going to talk about chipmunks anymore. It kind of makes sense because they go into torpor for winter. They're not really, up and down, active, inactive. They're mostly just, kind of checking out for the winter. So it kind of makes sense that we saw low activity from them. But the thing that I think is really interesting here is that equally likely to be consumed to the sugar maples are these invasive honeysuckle seeds, right? But only the Paramyscus are eating them, right? So the red squirrel's totally ignoring them, possibly because a red squirrel is much larger than a mouse, and so a tiny honeysuckle seed is less of interest to it. But here, this evidence would suggest that at least during the winter, deer mice are kind of 50-50 going after the native sugar maples as well as the invasive honeysuckle seeds, right? So sort of equal preference in the mice between these 2 food sources, and nobody likes hemlock seeds, right? So hemlock seeds get a free pass, but unfortunately they're facing stress in a lot of other ways, right? So it's good that the rodents are giving them a break. Okay, so just to sort of wrap up the winter portion. So snow, its role is really complicated, right? We're seeing a less reliable snowpack already characterizing our northern temperate forests. And we're seeing that these rodents actually have different preferences of what they're eating during the winter, but they're also showing different responses to that habitat structure, right? So understanding how winter climate change is going to alter the winter ecology of these northern temperate forests, we kind of need to account for these different species, idiosyncrasies, I would say, right? So it's a little bit about winter. I'm going to talk to you now about some stuff that goes on in fall, right? So sort of, going backwards in the seasonal progression of things. So everybody loves fall, right? We all love the colors popping on the leaves. But one of the things that we're seeing increasingly is like as these habitats get invaded by non-native shrubs, these shrubs actually do something quite a bit different with their leaves, which is so buckthorn, honeysuckle primarily is what I think about. They exhibit what's called this extended leaf phenology. And there's a big sort of comparative study in Nature a little more than 10 years ago that basically showed that it's kind of a consistent pattern with many invasive species. They actually leaf out early, and their leaves stay on like 2 to 3 weeks later in the winter, right? So they basically get a longer window with their leaves on where they're photosynthetically active. And for a while, sort of the end of the story was that, okay, so this is just a competitive advantage over the native trees and shrubs. They're able to do more photosynthesis. I think there was this study was looking at buckthorn photosynthesis, and something like 35% of its annual carbon is fixed in that week or two that it leafs out early. So it's really substantial from a physiological perspective. And that was kind of like, okay, that's sort of the be-all end-all about extended leaf phenology. It's about carbon fixation. But one thing that increasingly people are thinking of is that actually changes the extended leaf phenology that we see in these non-native plants, changes what the habitat looks like for consumers like rodents, right? So again, if we think about sort of the normal conditions, quote unquote, if you have a deciduous canopy, then sort of as the seasons progress, the leaves turn colors and fall off. You're basically opening up that forest floor canopy. The amount of light reaching, the proportion of sunlight that reaches the forest floor kind of goes up and then stays there, right? Whereas a coniferous forest, it's going to be more or less stable throughout. And so the point that I want to bring up here is that when you have this habitat structure change and you have increasing essentially visibility down to the forest floor, what this means is that the fall historically is a very high risk time to be a rodent. It lets the rodents sort of be more susceptible to their predators, things that are hunting them. But where we have extended leaf phenology, the sort of new normal in invaded forests, that window of high risk is essentially delayed, right? So now you have a much riskier, or much low risk, November and December as these leaves stay on earlier, right? And so I'll talk about this study in a minute, but just to sort of use it as context, this is actually looking at hard data on that. So the proportion of light penetrating down to about 0.5 meters on the forest floor. And you can see that we had a series of sites. This is at the UW-Madison Arboretum. Some sites that were invaded in red here. So lots of buckthorn prevalent in that habitat. And then an equivalent number of sites where we removed buckthorn experimentally, right? And so removing that buckthorn, yes, it makes a big difference sort of year-round, but the difference here in the summer is actually quite minimal, where the difference in the spring and in the fall is pretty pronounced, right? It's not just like a linear increase. It's definitely quadratic in that it definitely changes the most in spring and fall. And so rodents are they tend to feel safe under these thick canopies, right? And so we're wondering, okay, does that actually change these trophic interactions? And we found Christina Bartowitz, who was a master's student while I was at Madison in John's lab, did a study where it showed that, yes, actually this does translate into changes in seed removal. So again, in July, where that habitat structure is pretty similar, there's actually no difference in seed removal by rodents. But in September and in November, as those leaves start to fall off and those habitat differences become exaggerated, that's where you see, in fact, lower seed removal where buckthorn is removed. So there's a change in habitat structure, it's exaggerated in fall and spring, and that leads to changes in trophic interactions, but only in fall, according to the study here, right? They didn't look at what was going on in the spring. And so one of the questions that I kind of picked up after Christina's work in the lab was like, okay, is this just because there are more mice filling into these habitats, or are there more weird behavioral changes, kind of like what we saw under the snow in the winter? And so what I did was I put out, among other things, a bunch of, again, motion-activated trail cameras, and essentially looked at like what was the activity budget of these two different habitat types, right? When are we seeing rodents active in the invaded site, shown here in red, and the removal sites, shown here in blue? And here you'll see basically the overlap coefficient, so basically how similar these two distributions are. So an overlap coefficient of 1 means they're perfectly the same. They're doing exactly the same activity. They've got the same schedule. 0 means they're completely distinct. And so in the summer, there's actually a reasonably high degree of overlap between what time of night, and this is expressed here as sort of like hours after sunset. So 0 would be when the sun set, and then going on over here would be sort of the early morning hours. So what we see is that actually in the spring and fall, there's much, much less overlap, and we see much more distinct activity patterns between those two habitat types. And so here you can see that invaded habitats the rodents are coming out sort of like first thing, and then they sort of trail off. Whereas in the removed habitats, they're sort of waiting until maybe like 3 to 5 hours after sunset, and then they're active, right? So similar thing in the fall. And what we found out, and I'm not going to show the data now, but I'm happy to talk about it later at lunch or whatever. This is driven almost completely by the moon. and what time the moon is rising and setting, right? So in the invaded habitats, no need to wait. The moonlight's never gonna like trickle all the way down to the forest floor. It's not that big of a deal. You can go out whatever time. There's essentially no rules. But where buckthorn had been removed, the moon tends to be rising somewhere like, 3 hours in or whatever. And so that's the time, or sorry, the moon is setting. And so you have to sort of wait for the moon to set and then go out, right? So essentially these windows of activity are much longer where you have buckthorn because it's obscuring the moonlight, right? So again, sort of my logical next step is again looking at, okay, how do these behavioral changes maybe scale up to the community level? And so I'm on my pre-tenure sabbatical right now. I'm not teaching in the fall, so I can be out in the woods, stomping around, flipping over rocks and stuff. It's great. And so what I'm going to show you, just again, super preliminary. I actually have 2 students out there wrapping up a November round of what I'm about to show you today, like in my absence. So like we're only just really starting to unpack the data and what they mean. But we wanted to sort of know, okay, how do these behavioral changes influence something like seed removal over time, right? And so the way that we're thinking about this is that our experimental forest at Hamilton is a really great place to do research because we have a lot of conifer stands, we have a lot of deciduous forest beech maple stands, and we have some really invaded habitats, right? Lots of buckthorn in it. And so we have these sort of 3 different habitat types, and we're expecting that based on this idea of extended leaf phenology and seasonal changes in habitat structure, they're each going to kind of look different in every season, right? So in the summer, they're maybe all looking similar, but then in November, we would expect that, okay, even the buckthorn leaves have maybe dropped by then, but the conifers still have a nice leafy habitat overhead. And in September, maybe the invaded forests look somewhat like the conifer forests, but the deciduous forests have already started to drop their leaves. So our hypotheses here were that we expect to see sort of a decline in the rodent foraging activity from summer into fall when this habitat is opening up. But we expect that this will be slower in the habitats with more conifers and more invasive shrubs because of these differences in leaf phenology. And the other thing that I'm really excited about to really strongly connect this to the community assembly piece is that we expect that if you look at foraging behavior in rodents, in the same time that you're actually measuring seed to seedling recruitment, is often a missing piece in these studies, we're expecting to see that where we have the most foraging activity, then we're going to have more seedling recruitment, essentially. So the main piece of this is essentially what we're calling a seed addition study. So you put a bunch of seeds on the ground, and you cage some of them, and you have a sort of fake cage with holes in it that lets rodents in, and then an uncaged plot as well. And you look at how the different seeds transition into seedlings over time. And that tells you something about the role of these biotic interactions. And then you pair this with a set of plots where you don't add any seeds, and you see what naturally comes up. And that tells you something about dispersal limitation, right? So this is really the community assembly piece of the whole thing. But at the same time, we're running these seasonal giving up density and seed removal studies to look at how things like seed traits or consumer behavior, like risk perception, might or might not be correlated with these seed to seedling transitions. Unfortunately, I'm not going to know anything about this until next summer. So maybe I'll have to come back again and tell you guys what happens there. But we're replicating this at 15 plots where essentially we have 5 habitat types of sort of each of these 3 categories: coniferous, invaded, and native deciduous. So I'll really quickly, because I want to sort of get through this, the first thing I want to point out is that there's actually, the whole premise of this was that there would be extended leaf phenology happening in these invaded habitats. What we actually found so far, and I'm showing you essentially, again, that proportion of light reaching the ground level plotted against the relative abundance of conifers on the left and buckthorn on the right. And so September here is shown in gold, and the November trial is shown here in red. And so this is a nice example of sort of that leaf phenology where you don't have any conifers and you just have the deciduous species, that there's clear separation in how much light is reaching the ground. That makes sense, right? So if you're in a deciduous stand in November, there's a lot more ground that's lit up than, say, in November, right? But what we see here is that, again, that's sort of consistent with this idea that conifers are blocking the light even late into the fall. I expected to see something pretty much identical to this, but maybe weaker with the buckthorn plots. But actually, we see nothing like this. So this whole study was sort of predicated on the idea that extended leaf phenology is a common and consistent thing that invasive shrubs do. But actually what we see is that where you have a greater relative abundance of buckthorn, we actually have more light reaching the forest floor, regardless of the season, right? And it's a marginally significant association there. It's not the super strongest finding, perhaps. But this is really interesting, and I was thinking about this. I was like, what the heck? Like, this is sort of antithetical to the whole idea of doing the study. And I was thinking about what these plots look like. And what we find in our particular neck of the woods is that these are old abandoned farms that have, reconverted through succession back into secondary forest. And some of them look like beautiful, secondary forests that you just think that you're walking around in the woods, right? And it's always been woods. But then these other buckthorn-infested habitats tend to have a lot more essentially feral apple trees that have been essentially left over from old apple orchards. So it's a mix of not just buckthorn, but also these old apple trees. And those apple trees are like 20 to 30 feet tall. They're really large, and those things drop their leaves really early. So it tends to be that there's this, buckthorn, it can grow as just a buckthorn thicket, but it might also co-occur with other species that then give you something less expected. Right? So it's this totally new phenomenon happening to us. And so far we've been able to process, again, the July and September seed removal samples. November, we're just about to collect them today. And what we see is that there are some clear patterns in what seeds are being eaten, regardless of the month. So American beech and white pine, those are the two highest preferred seeds for our rodents so far, kind of consistently. And then I'll point out here the honeysuckle and buckthorn. These are, they're not the most preferred species, but they are still being consumed, in pretty high numbers, so like 60% or more, right? And then, so the last piece that I'll show you from this study is essentially looking at, trying to link this back to the seed removal part, right? So a similar x-axis here, so relative abundance of conifers versus how many seeds are removed. And if you plot it in terms of the conifers, again, you don't really see a clear, strong pattern here. Regardless of whether you're in a deciduous or a coniferous forest, seed removal is just high all the time. But if you look at this again in terms of the relative abundance of buckthorn, in the September trial, shown here in the gold, we see actually a pretty strong drop-off in these sites where we have more buckthorn. And again, this is kind of opposite what we expected, because we thought, okay, buckthorn, lots of animals behaving weirdly, active all the time, there's going to be more seed removal. But remember, in our buckthorn stands, there's actually more sunlight reaching the forest floor. So at first I was like, oh my gosh, this is really weird, low seed removal in buckthorn, that doesn't make sense to me. But this is actually consistent with that sort of behavioral rule that where the habitat structure is really open, we actually see less seed removal, right? So that is actually consistent. It's just expressing itself in a different way because the buckthorn is doing different things, right? And so I'll wrap up here on my last slide here, looking at sort of the next step of these data. So I mentioned we have sort of so one piece that I'm not showing you here is, in addition to just counting the number of seeds left over, we're doing the same trash cam setup here where we can actually see which individual rodents are coming in and foraging in the different habitat types. Processing those data takes the longest time of anything that we do in our lab. So we're still working our way through the July samples, let alone September or November. One of the things I mentioned was that it's hard to know which of the two Peromyscus species in our forest are actually coming in, and you don't know anything about the individuals that are actually foraging in those buckets. But one thing that I'm really excited about is that in addition to the trash cam ring doorbell setup, I'm also adding into a subset of these sites here what I'm calling easy pass for mice. So when we go out and we monitor the small mammal community, we give them all pit tags, so an RFID tag with a unique serial number so you can tell them apart. That's essentially the mark part in the mark recapture. What you can do with that is you can actually throw down an antenna that can read RFID signals around a bucket of seeds, like our trash cans. So here's an example of that. So we have 6 sites where we deployed these easy pass for mice RFID antennas. And so they're sort of color-coded over here. You don't need to know like what those mean or where they are. But we can actually see different individuals coming and going on different nights at different times of night, right? And so like, that's pretty cool. Like you actually get kind of like a traffic record of who's going in and out. And if you sort of blow that up on that first night, one of the things that's really exciting, you can see, okay, there's actually a lot of activity in some of these, and I can say, okay, if I match this up to our trapping records, that's a Peromyscus maniculatus, maniculatus, maniculatus. Oh, but this one's a Peromyscus leucopus, right? So we can actually go into these records eventually and tie that back to the photos that are time-stamped according to like when this Peromyscus leucopus went into the bucket, and actually match which seeds did they go. Did they go and eat white pines? Are they going to eat buckthorn? That sort of thing, to look at these sort of like perhaps cryptic or often obscured sort of associations between different species of rodents and what they're eating, right? So invasive species, they're monolith. They're commonly eating a lot of these invasive seeds, actually, which is really interesting, because one reason why we think invasive species might be invasive is because nothing eats them. They're allowed to just sort of, have an enemy-free space. That's not clearly the case with their seeds, at least. And we also have this weird finding that this extended leaf phenology isn't a key driver of rodent foraging activity everywhere. It was in Wisconsin and in other parts of the world. In central New York, not so much, because these weird apple orchard savannas that you get to become reinvaded with buckthorn. And so the next step that I'm really excited to unpack is sort of looking at these inter and intra-specific differences by linking this new behavioral data with the cameras, with the RFID tags, to sort of link that to exactly like evidence of what they're eating on a given time. So that's what I've got for you today. Thanks so much for listening, and I'm happy to take questions. I think we've got about 5 minutes or so. So, yeah.
Okay, we have time for some questions, and I'm going to try and repeat the question to make sure that our audience online is able to hear it. So you can call on who you talk to.
Sure, yeah. Thank you. Yeah.
I got thinking a little bit about predators.
Mm-hmm.
I don't know a whole lot about rodent predators, but I'm wondering whether there are different rodent predators that behave differently in terms of how they're doing their foraging, and whether you've thought about trying to layer that onto this argument.
Yeah. It's definitely something that I think of. There tend to be, as I see it, sort of like three main groups of those predators, which would be raptors, and then canids, so things like foxes and coyotes, and then mustelids, so things like, weasels. From other trail cam monitoring setups that we've had, we occasionally get coyotes coming through. I have not seen a fox on our trail cameras since I've been there. And so I don't know if they're just, if our experimental forest habitat is too fragmented, because the land it's about the only contiguous stretch of forest in our part of the woods, right? So there's just not a lot of connected forest landscape. So I would bet that raptors are the primary one here, but I would love to think about that because there's different, olfactory cues versus visual auditory cues. Fascinating to unpack that. And I would love to keep thinking about that, but for now, I guess we're trying to simplify it to get off the ground. But it's something I would love to think more about. And perhaps if you baited in those canids, maybe you're more likely to see them or something. But yeah.
Let's repeat the question after.
Oh yeah. Thanks. Yeah. Mm-hmm. Yeah, great. So the question was, how is snow impacting these different guilds of predators, right? So like raptors versus mustelids versus canids. So that's a great question. The only thing that's going to go into the snowpack really to consistently eat a rodent would probably be mustelids, specifically like things like American martens that are really specialized at diving under the snow, using that. They've got that sort of like small, long body that can like use that tunnely system and get in there. The weird thing about snow, especially in the context of the data that I'm showing you so far, is since I've been at Hamilton, the first year when I didn't do any research because I was still just like figuring out the teaching side of things, we had a normal winter. The last 2 winters, we had almost no snow until April. And so we launched some studies to look at Subnivean interactions, And then those data were just kind of like, it never snowed. Like, so I feel like in some ways we're like 20 years too late in like getting some of these cool projects off the ground. So we don't really know like how snow or loss of snow would, to your point, sort of influence the importance of one guild versus the other. But I think that would be really fascinating. And yeah, I could think of we can maybe talk later about different ways that you would get into that. But it's a really cool idea to think about those different groups of predators, yeah.
Okay, we are going to move into the lunchroom for the next stage of Dr. Guiden's visit, and please join us there. Certainly, if you have any more questions, he will be here for the afternoon, and approaching him in the lunchroom is a good way to make sure you set that up if you don't already have a schedule.
Yeah.
And
Thank you.
thank you.